and I would like to welcome you officially to the very first PataFEST Citizen Science webinar. Oh, wait a second. I wanted to share my screen and it didn't work, of course not. Okay, second try. Can you see the screen? Perfect, thank you. All right, so I will, there might be a camera issue. I hope that people who will look at the video later will still be able to see the speakers. Anyways, I'm Leo. I work for the European Citizen Science Association, and yes, we're going to have in total 8 webinars where we're going to focus on PataFEST as a project, as well as on citizen science as a tool to include citizens in agriculture and food. For this, I'm happy to briefly introduce the next upcoming webinars. So we will have one taking place in, so this is the first one, Introduction to Citizen Science and to PataFEST. We will have the second webinar on testing biofertilizers coming in April, another one on platforms for agri-food citizen science projects, and another one on potato DNA extraction. I will go more into detail about webinar 2 and 4 during a later presentation this webinar, but just to provide you a broad overview. However, I would also really like to introduce our first speaker, which is Christian Ghidelli. He's the project officer, or project coordinator for PataFEST, and he knows everything there is to know about the project. So I think I will give you control, and hopefully you can do your presentation. Okay. Thank you.
Thank you, Leo. Let me try if I can move. Okay. Thanks, Leo, for organizing this webinar and to giving us the opportunity to present this promising project, PataFEST. Briefly, I would like to introduce myself and Funditec. We are a Spanish technological center located in Madrid, and I'm working as a principal researcher in the agri-food department. Just for your information, in PataFEST, three main departments are participating, like agri-food, digital transformation, and advanced material. Beside me, Maddalena is the project manager of this project. Okay, let's go into the background. Potato is one of the most popular food in the world, and for instance, in Europe, the potato production is more than 50 million tons, with higher production in Germany, Poland, France, and Netherlands. In South America, the cultivated potato are widely distributed in the Andean region, with more than 4,000 potato varieties and more than 100 wild species. However, potato is very vulnerable to a variety of pests and soil-borne pathogens, resulting in significant food and economic losses within the potato industry. In view of this scenario, the European regulatory framework for the potato imports and production set strict requirements. For example, the European and Mediterranean Protection Organization, the EPO, established phytosanitary measures for seeds and wild potatoes to prevent the introduction and spread of quarantine pests. Regarding the challenge in the potato cultivation, well, we can define that the most important pest in potato plant is the bacteria Candidatus liberibater solanacea, which disease is known as zebra chips. The zebra chips generate like brownish shade in the flesh of potato, as you can see in the second pictures. And so, having the disease, the potato tubers cannot be marketable and also cannot be suitable for the processing, like to produce French fries, potatoes, etc. The bacteria is moving, or well, the psyllids that is responsible of the propagation of bacteria from a plant to another plant. And the psyllid is an insect, okay? It's called Bacterisera cockerelli, that is shown in the first image. In addition, the reduction of the use of chemicals in field, okay, synthetic chemical in fields, as required by the Green Deal regulation, will generate a high incidence of post-harvest soil-borne pathogen, like Fusarium, Colletotrichum, and Mythosporium during the post-harvest storage of potato tubers. Okay, so I cannot move. Okay, so some information, numbers regarding potato, regarding this project, this PataFEST. The overall budget is around 6 million, and with a duration of 48 months, in which 18 partners is participating from 9 different countries, including Europe and a country abroad that is Ecuador. The PataFEST project is divided in 3 main tasks. The first one will be the molecular characterization of pest and potato by including molecular bioassay and the bioinformatic approaches in order to understand how the pest is spreading, and also to select resistant potato varieties against the plant pest and the soil-borne pathogen. The second one will be to provide solution for the pre-harvest diseases, like developing image analysis digital tools and artificial intelligence algorithm for the early detection of the disease. As a prevention tools, bioinsecticides for plant application will be developed, and for the plant soil management, natural and sustainable biofertilizer will be developed to improve the soil health of potato crops. Regarding the last task, it's to control the post-harvest diseases. Sensor to detect volatiles will be developed for the warehouse, potato warehouse, and as a prevention solution, biocontrol formulation coatings will be developed and combined with control atmosphere to store potato during the post-harvest period. Okay, all these technological solution will be integrated in a pest management strategies in which validation field will be carried out in Ecuador and in several places in Europe, and same things for the warehouse trial validation under real condition here in Europe. Okay, the project also will include the engagement of citizens with several activities as practical workshop, webinars, citizen science campaigns. In addition, a multi-actor participant platform will be created to share knowledge, experience, and ensure economic sustainability of European potato production. Okay, some achievements, some main result of that we obtained within this almost 2 years of project. Well, results related to the molecular characterization of pest and potato. More than 100 novel resistant genes and around 700 non-resistant genes have been identified. And around 350 potato varieties have been tested against Bacterisera coquerelli, that is the psyllid responsible of the Candidatus liberibacter disease, and more than 200 resistant genes varieties have been evaluated against the key soil-borne pathogen in the post-harvest storage. Okay, regarding the pre-harvest disease solution, a massive number of images are being collected as data sources for the development of the predictive digital platform. Regarding the treatment for plant, bioinsecticide based on a mixture of clays, essential oils, incorporated into a wax-based coating, showed higher mortality rate in 25 hours against aphids. And finally, for the soil management, seashell-based slow-release fertilizer have been developed, and the trials in potato greenhouse have been setting up to evaluate the pest pathway of entry and drivers of spread under future climate scenarios. Finally, the last result about the last task to control post-harvest diseases. Main volatiles have been detected, okay, to develop the portable sensor, and also the materials that will be used to produce this, to manufacture this sensor for the warehouse conservation potato of the potato. And finally, for the prevention solution, killer yeast and the enzyme have been characterized, selected as biocontrol agent that will be incorporated into a coating formulation based on a mixture of polysaccharide and lipid base to control pathogen spoilage and maintain the quality of the potato tuber during the storage. This solution will be also combined with the control atmosphere condition. Well, finally, just to encourage you to follow the news and the project updates of PataFEST in Twitter, X, LinkedIn, or YouTube. And so many thanks for your attention.
Thank you very much, Christian. I will check in the chat if there's any questions. Otherwise, if you would like to know more about it, I know for some of you it might be very technical, so also feel free to ask questions just to simplify it. So I see Marie, you have your hand raised. Please go ahead.
Hello, thank you. I do have a question. First of all, it's really remarkable that within 24 hours you can see a big reduction of the aphids. And what about like larvae or other insects? Did you notice? Because like it happens that we use neem oil from India, and it keep away the aphids, but also other animals, small insects.
We be focused on aphids because, as you maybe know, that we cannot work directly on the Bacterisera coquerelli since it's a quarantine pest. Okay, so we cannot manage here in Europe trials with this kind of psyllids. Okay. Therefore, we use cotton pitch aphids. Okay. No. Yes. Well. No. Pitch aphids, it's called, I guess, as a model. Okay, to validate at lab scale our solution. So, and we be focusing only on that. We didn't try any other, evaluation about other kind of insects. However, we are going to start a validation in the coming months. And so we are going to do a treatment of aphids against aphids here in Europe, but also in Ecuador. Okay. Firstly, in greenhouses against Bacterisera coquerelli. I guess that we are going also to evaluate the impact of our solution here in Europe in field, also against other kind of insects. But for firstly, because the main issue is aphids for potato crops.
Actually, I'm talking about good insects. I'm not talking about other bad insects.
Yeah. I know that maybe our solution can be also antagonistic against something that is good in the field, and is something that we are going to evaluate.
Cool. Thank you.
It's already included, okay? Because we are going to put traps or something like that, and we are going to evaluate the effect against not only the aphid, but basically we are focusing on that. But it is something that is included and we are going to take in consideration.
Wonderful. Thank you both very much. Also, feel free to continue asking questions, or if you are interested in joining any of the activities, even if we don't mention them as direct citizen engagement activities, but you have ideas of how you would like to contribute, we're very happy to hear about it. So yeah, again, we will, if you have any other questions, please feel free to also write them in the chat, and we can go through them at the end of the webinar. But until then, I'm happy to give the floor to Anna. I will switch, and hopefully you're able to share the screen now. Anna is a colleague of mine at ECSA, so she's also very much experienced in citizen science, citizen engagement, and will tell us a little bit more about the Open Soil Atlas project. Anna, please go ahead. Oh, wait, I need to mute myself first.
Okay, sorry, I needed some time to find the button. Okay, so I'll go back to the previous slide. Okay, here we go. Hello, good morning, good afternoon, everybody. Indeed, yes, my name is Anna Verones. I work at the moment for the European Citizen Science Association together with Leo. And I will be talking today about the Open Soil Atlas project as an example of citizen science application in the agri-food sector. I won't be talking so much about potatoes. We are moving a bit away from that, and the project is more focused on soil, and more specifically urban soil. Leo asked me to give a bit of a definition of citizen science. As we have many definitions in the literature, I picked 3 of them, which I found being comprehensive. The one being from the European Commission in 2014, which says that citizen science is the general public engagement in scientific research activities when citizens actively contribute to science. So it like stressing the way that citizens are involved. So also diversifying from science communication or public engagement and attributing a more active and meaningful role to citizens. In 2015, ECSA defined the 10 citizen science principle, and in the first one saying that citizen science projects actively involve citizens in scientific endeavor that generates new knowledge or understanding. And a final, more recent definition is from the Impetus project, which also similarly said that citizen science projects aim to contribute to research, produce new research-based knowledge, and their activities are carried out by participating citizens. So you see the common points of involving citizens in scientific research, as well as producing and generating research-based knowledge. And that said, we can look at Open Soil Atlas as an example of a community-based citizen science project where citizen scientists had the chance to map, reclaim, and remediate urban soil. It ran from March to August 2021, and it was funded by Action, Horizon Coordination and Support, Action supporting citizen science practitioner in both through training program and also with financial support of 20,000 euros. Moving to the next slide. Okay. I will today talk about the project more in general and why we decided to use citizen science, how we engage the community and citizens, and also the soil test protocols that we defined and adopted, and shortly about the results that we got. Open Soil Atlas came from the Fell Food Forest community, which was a urban gardening, food forest and permaculture project that was born with the vision of transforming what you see in the picture, the Temple of Erfeld in Berlin, into, not the entire area, but establishing and growing a urban garden there as a space for education, experimentation, and community building, all around the topic of sustainable agricultural practice in cities. The area is an abandoned airport. It used to be an airport and a space for military parade in the very past. It's not being an airport since the last couple of decades. It's now more a space for recreation and a space for nature. But considering this, it was very important for us to also work on the topic of soil remediation and soil care. And as we wanted to plant edibles on this area, we really needed to get good information about the soil, so whether there were contaminants, then also the fertility of the soil and many different aspects. And here we encountered the first barriers, being it, wait, sorry, slides go a bit slow. Yes, so the barriers that we encountered were both a lack of information about public urban soil. This data and information have been collected, were available, but were not available publicly, even if it was about public soil. And also a low public awareness about the importance on the soil, both on the side of policy making, and also among the society at large. So we wanted to address these, and we decided, okay, let's start generating this information, collecting them and making them available, and at use for also more sustainable city planning decisions. That's why we decided to use citizen science to get agency in this process and back up our statement with scientific data. And our research question was about how do external factors and different land uses affect the quality of the soil, and which management practices are most efficient to regenerate the soil. So we came up with Open Soil Atlas as a soil testing co-learning center. So we created a website where you can find a lot of information about soil, explained in a very simple way, and also guidelines about how to test the soil in your garden, close to your place, or wherever you want. Citizens would engage by testing the soil, collecting this information and uploading them on a platform with a GPS location, and therefore generating the soil map that you can see also in the picture. This information was supposed to inform city planning and urban horticulture practices, and it was also supposed to be replicable and scalable. Indeed, the tools can be already used in different areas and for different purposes. Here I have a slide about how we were internally structured. So we were divided into 5 main teams, one being impact assessment, one being graphics and design. Then there was the work of research and development, where also the, let's say, scientific profiles among the team were more involved, so designing the testing protocol, the tools, and the trainings. But then also a lot of resources went in engagement and communication, so both on social media for the public at large, and also more targeted outreach and networking. And this brought us to establish a very nice community of soil enthusiasts from different backgrounds, having several partners, cooperators, as well as experts and researchers, and the Berliner city administration. And of course, a lot of citizens who engaged and were the essence of the project. Talking about how we engage the community. And maybe first of all, why we engage the community. That started from the idea of the correlation between healthy community and healthy soil. So we've seen the importance of making people aware about what the soil is, why it's so fundamental for us, and why we need to take care of it in order to empower them to also take care about themselves and explore this connection. Citizens engaged actually in different phases of the project. We were a community-based citizen science project, so the engagement was very strong since the beginning because it was promoted by the civil society as well. The idea was indeed to democratizing soil science and enhance soil literacy across the community in a very simple and accessible way. So also trying to promote equity, diversity, and inclusion. So we engage people from different districts, from different, let's say, categories, and it was very nice to see how people that usually wouldn't have access to such scientific soil-related information, but could go deeper into the topic and explore it together. Here a bit of the timeline of the project or how the engagement program happened. So the project was a 6-month project. We were initially planning to have monthly soil meetups, so training moments for people to learn about how to test. And the first 2 happened online because of Corona. And the idea was that people would get this information and then they would go out and test the soil and collect the data. However, we realized that the process was a little bit demanding for them to carry it out by themselves. So they needed maybe a little bit more guidance and support. So we somehow needed to meet and be together with them to walk them through the process. So we started to go on the street, and you see the soil meetups intensified in the summer months. And many people participated. Some of them also participated more than once and got further engaged with the project team and became themselves soil ambassadors. So people who were trained and motivated to get other people involved. The process was intended to have also this snowball effect in order to get always more people engaged. I will show you briefly what kind of information we have collected. I would like to start from the toolkit. So we first of all developed this manual that maybe you can see from the picture, which is plastified and contains all the guidelines to carry out the test and could be brought by participants on site during the testing procedure. And in the first page, you see the toolkit. Maybe you can't see so well from the picture, but the toolkit was very basic. So we had, they needed water, a shovel, vinegar and baking soda. I will tell you later why. Some small plates, spoons, and I believe that was all. And that was intentional to allow a lot of people to participate because these are tools that almost everyone has at home. And of course, the quality of the data might be lower than what you would get in a laboratory, but on the other side, you have the possibility to engage way more people than what you would engage in a laboratory. And looking at what was collected. So here I put also a little bit of pictures on the right side, and on the left side, you see the manual that I was showing. So the first information was about the land use. The second and then, sorry, still from the land use. So to which category, for example, green, urban setting, agricultural area, or forest. These were adapted to the local context where we were using it. Then participants looked into the anthropogenic impact in terms of trash that they would find around the area. And make observation about the soil erosion. Then they would start digging, and a 20 cubic meter hole. And while digging, they would count how many earthworms they identified, as well as any other biological activity. Once they have digged the hole, they would look into the soil profile and tells whether there is topsoil, and what is the color, whether it differs from topsoil to subsoil, and also indicate what the color is according to the scale here on the manual. Then we would look into the soil texture through the ribbon test, and also through the jar test, which was one of the funniest moments where participants would shake the jar with some soil, and then they would let all the material deposit and observe how differently they layer, and also the organic material floating on top. And at the end, we were also testing the pH. Here again, we were not using the stripes. We were using only vinegar and baking soda, and looking at when mixed with the soil, whether a reaction occurs or not. So it involved also some, how do you say, sensorial observation and interaction with the soil. Yeah, that was the information collected. I wanted to say something about, the results. So we could really observe, okay, first here you can see the map that we generated. I think at the end we got around 130, 150 observation distributed across Berlin, and also sometimes only in one street, so a more like targeted counter mapping activity of the street greenery. And they are available on EpiCollect. There is the link here in the slide, both as a map, but also as a data table with all the detailed information about each observation. Yeah, we could, so what are our results, what we learned from the process? So definitely we could observe how the quality of the soil varies according to the land use of the area and the level of maintenance. Mostly, for example, in urban gardening, where a particular attention is put to the soil, you could really see the impact of it in terms of fertility, in terms of capacity to drain water. So that was very visible and nice to experience. And maybe more on a social science perspective, also we could really observe how soil is an important element, or could be an important element of ecological and social unity and cohesion. So that was also very nice to observe, looking at how people engaged and connected through it. Yeah, here it's about other data platforms that can be combined with the data we collected. For example, one about street trees, or one about how paved the soil is across the city. So maybe they, as data, they can then be also stronger for further impact at the policy level. And that said, I took a little bit more time than what was planned, but thank you very much. And yes, I pass it back to Leo.
Thank you very much, Anna. I will check real quick if there's any questions or comments in the chat. Again, in the meantime, if you're interested to participate or to if you have ideas of how to connect the two projects, for example, feel free to share them as well. If there are some questions, I mean, I was just curious, did people exchange, let's say, strategies on how to improve soil health? So since I'm also based in Berlin, I know the soil is not very good, or I assume it's not very good. So was there any communication on that?
Yes, definitely. And many events also happened in community gardens, like urban community gardens. So there was also the experience of the gardeners coming up and giving a lot to the participant. But it was very like both directional exchange, and participants were asking and learning also what they can do in their gardens or also on their balconies if they want to work with soil there. So there was a lot of knowledge exchange going on and a lot of knowledge generation as well.
Lovely. Oh, that's so great to hear. Yeah, thank you so much, Anna. I think that again, if people have more questions, feel free to write them in the chat. We can also have a little bit of a discussion later. Is this toolkit still available, for example?
Yes, I can send the link here. And yes, it's still available. And there is also my email address. No, the email address of the project in the last chat. And if you want to use it or look at it together, feel free to reach out. But I will send the link to the online learning center now.
Thank you. I also see that we have another raised hand. Maria, would you like to say something?
Oh no, well, I guess once you send the link, I will look at it. Yeah, I was wondering if that manual to analyze the soil, it was only in German or it was in English, or if you did it in several languages. I guess if you only did the study in Germany, it would make sense that it's in German only, right?
We had the German version, but actually it was we have German and English.
Yeah.
We started with the English version and then made it in German for the local community.
Yeah.
Yeah,
But
so these are the two languages that we have.
yeah. Okay. Then forget about trying to do something like that in Spain or something. But yeah, it's very initiative. Very nice.
Yeah, it would be nice to translate it also in other languages, actually.
Yeah, because I think it's a very good way of mapping the soil using citizenship science. If, for example, you send this activity to schools, possibly the science teacher will take the children to the local backyard and analyze the soil and can send you back the data.
Yeah.
Yeah, maybe it's a follow-up project. I mean, it also links a little bit with the question from Beatrice, which is how could this be scaled up in other countries? So was there an idea on how to do this already? Or have you thought about it?
Yeah. So I see it separate. So there are a lot of urban gardens in Berlin. It's very vivid, vital activities. However, they all encounter huge challenges from gentrification, and they are continuously moved from one area to the other. So it's a lot of motivation, while a lot of challenges as well. This could be scaled up in other countries. So in our project time of 6 months, we focused on Berlin in order to have a good body of information, at least for the area addressed. However, it applies to everywhere actually. Like maybe you also have more information than me about that. I done it mostly in Berlin, but it's also some general information that or general testing that can be conducted almost everywhere. I see the part about the land use, maybe the one a bit more context specific, but for the rest, it can be used. We would need to establish some connection outside of Berlin and try to outreach more. But yeah, this didn't happen in the project phase, but yeah, we are not funded at the moment, but it could be very interesting to do it as well.
Wonderful. Thank you so much. Also, since I'm not saying we're running out of time, but I also wanted to briefly maybe introduce the ways that we want to incorporate citizen science within the PataFEST project. So as you know, since Anna and I are colleagues and we're both working for the European Citizen Science Association, we're thinking of ways that we can involve citizens. So similar to the Open Soil Atlas project, PataFEST also is dedicated to involving citizens in the research project. So now I'm trying to basically combine the two presentations we just heard by explaining the activities that we have planned within Berlin. We also have partners from Spain, so they can also answer questions on similar activities happening there a little bit later, hopefully. Yeah, I'll start with the first one. So our first activity is a workshop where we're inviting gardens to test biofertilizers. Basically, PataFEST partners have developed a biofertilizer that's based on mussels, so mussel fish byproducts. And the idea is for anyone growing potatoes really to make a comparison between what it's like to grow the plant with a biofertilizer and then compare to what happens if you use conventional fertilizer or none at all. So we're inviting gardens who are interested in experimenting with different fertilizers or different ways of growing potatoes to join our workshop. We will have a webinar where we will explain more the details of what the biofertilizer is composed of and how the workshop is going to look like. But just to give you a general overview, if you're interested in joining this activity, any future webinars, or the activity I will explain next, then feel free to scan the barcode in the corner. I also kept it on the same slide, so no worries, it's the same one. And this is another of our activities, which is participating during the Long Night of Science. So since ECSA, or the European Citizen Science Association, is based in the Natural History Museum, we reached out to their molecular biology lab, and they agreed that we can invite anyone growing potatoes to bring in their own potato plants. So maybe not the entire plant, but you can just bring in your leaves, and then we will do a DNA extraction and PCR sequencing. So similar to what Christian said in the first part of his presentation, we want to see how you can actually assess whether or not a potato plant is resistant to certain diseases or not. And with this, we're happy for anyone who is planting potatoes this year to sign up for this event as well. And as always, if you have any questions, feel free to ask them now, feel free to reach out to us. And that's it from my side. And as Christian has already said, we're very happy for anyone who will follow PataFEST on social media. We also launched a podcast, which is called Potato Talks. So you see that Christian can tell you more there about the PataFEST project. Yeah, and if you want to rewatch this webinar or any of the future webinars, they will also be available online. And wait, I'm going to check if there's any other questions in the chat. Just many congratulations on the Atlas Soil project. So I hope we can use this as a inspiration for any PataFEST activities. How are you going to do the PCR to give the results to the citizens the same day? We have the equipment, so it will take a while, definitely, but we will do a PCR sequencing that should take 2 hours. Our partners at the molecular biology lab are currently doing the first tries. And yeah, it's it's possible. I can also give you more details on this, Maria.
No, it's very nice. It's very nice.
Yeah.
Yeah. I was wondering which kind of PCR was so sequencing was so fast. Probably is yeah.
Yeah, I can't give you the direct name. Sorry, that's where my expertise also stops. But if you're interested, I can send you more details on it later.
Yeah, it's very cool activity indeed.
Great. So are you joining?
I cannot go to Berlin, but
Oh.
I will.
But there's, again, as I said, we have Spanish colleagues here as well, so there will be a similar activity.
Yeah, I can send the potato leaves too.
Perfect. So is there any other questions? Okay, oh yeah, Victoria is the partner from Spain who's responsible for it. All right, then I'm going to now stop sharing the recording and then we can have a little bit of a less formal chat, let's say.